If a vial of your blood sits on a warm laminate counter for just an hour, your physician might diagnose you with a rare, potentially fatal genetic disorder the very next day. The diagnosis is a false flag. It is an illusion caused by a single microscopic molecule in that blood spontaneously changing its shape. This is L-aspartic acid, or aspartate. Within the human architecture, this molecule operates as a chemical metric, tracking nitrogen stores and mitochondrial fuel states. Its core biological mandate is to link structural carbon availability directly to systemic energy output. This amino acid converts those metabolic signals into central cognitive drive, providing the materials needed to wire a structural memory layout. To understand how one molecule manages this, we must map its journey from its rigid molecular blueprint to its role as an energy smuggler, its capacity to trigger rapid brain death, and the diagnostic traps that make it so difficult to measure. The behavior of this single molecule dictates the threshold between stable synaptic plasticity and catastrophic neurocollapse. Biologists classify aspartate as a non-essential amino acid. This means the human body possesses the complete internal factory required to manufacture it from scratch. Aspartate forms a short dicarbolic chain, a microscopic barbell with a single spacer. This short spacer makes the molecule exceptionally rigid. While longer molecules bend, aspartate stays completely stiff. At normal blood pH, the barbell's ends carry a strong negative electrical charge. This combination of an unyielding backbone and a powerful negative charge turns aspartate into a microscopic electrostatic anchor. Inside the body, it uses that negative charge to grab positively charged amino acids, forming an ionic attraction known as a salt bridge. These salt bridges are structural linchpins. They physically lock massive, complex protein ribbons into the precise 3D geometries required to function. Because of its intense electrical power, the body heavily restricts where this molecule can travel. In circulating vascular fluid, free aspartate levels are kept to a bare trace, usually between 1 and 10 micromoles per liter. Inside the cells themselves, however, the concentration is maintained at levels up to 100 times higher than in the blood. This massive gradient creates an environment of wild animals held in a cage. Aspartate's immense structural power is highly effective, but it must remain strictly contained to prevent systemic chaos. The first place we see this power harnessed is inside the cellular power plants, the mitochondria. Their inner membrane acts as a solid brick wall, completely impermeable to a large energy-carrying molecule called NADH. Because that barrier is locked, the cell cannot push this heavy energetic package through the front door. To bypass security, biology developed a transamination exchange. It strips the high-energy electrons off the bulky NADH package. The cell needs a vehicle to transport that separated energy, and aspartate serves as the designated smuggler. Aspartate takes on the excess nitrogen and crosses the barrier from the inside out. This revolving door exchange ensures the high-energy electrons can finally enter the power plant. This indirect smuggling ring, the Malate aspartate shuttle, is mandatory for the cell to maintain maximum ATP generation. Beyond smuggling fuel, aspartate also takes on a secondary synthetic role. It provides the raw physical scaffolding required to build human genetic code. The molecule dismantles its own structure, sacrificing its carbon and nitrogen atoms to construct the pyrimidine and purine rings of DNA. If the body lacks this rigid dicarboxylic acid, the mitochondrial power plant starves, and physical replication of new cells grinds to a halt. When we move from metabolism into the central nervous system, aspartate shifts roles, acting as a primary excitatory neurotransmitter. Its negative electrical charge facilitates binding with specific NMDA receptors on the surface of the neuron, initiating the localized calcium influx required for synaptic activation. This opening permits a brief, tightly controlled pulse of calcium to flow into the brain cell. That pulse of calcium acts as a signaling trigger. It activates enzymes that physically remodel the synapse, driving the structural rewiring we recognize as memory and learning. To keep this signal from overwhelming the brain, astrocytes act as cellular janitors. They rapidly vacuum excess aspartate out of the synaptic gap. There is a massive biological vulnerability. These astrocytic vacuums run on EAAT clumps, which are strictly dependent on the continuous supply of cellular ATP. If a patient suffers an ischemic stroke or a severe traumatic brain injury, the tissue loses oxygen. The ATP energy grid collapses almost immediately. Without that energy supply, the astrocytic janitors are instantly paralyzed. The cages break. Intracellular aspartate floods the synapse in an uncontrolled wave. Without active pumps, it binds to NMDA receptors, jamming the channels permanently open. Instead of a measured pulse, a river of calcium violently floods the vulnerable neuron. This sustained calcium spike hyperactivates the enzymes meant to remodel the synapse. In overdrive, they begin to physically digest the brain cell from the inside out, triggering rapid neuronal necrosis. This excitatory mechanism also creates a dietary vulnerability. Over-consuming artificial sweeteners that break down into free aspartate can overwhelm peripheral NMDA receptors, causing sudden systemic flushing and tremors. Brain death during a stroke results from the accumulation of its own signaling molecules, which initiate an uncontrolled cascade of neuronal destruction. When a crisis occurs, emergency medicine relies on tracking specific molecular distress flares to monitor the extent of cellular damage. This introduces a critical blind spot. On a standard metabolic panel, doctors often confuse the raw substrate with the processing enzyme. Think of the free aspartate dissolved in vascular fluid as raw lumber. It is the simple amino acid building block. If a lab reports an elevation in this free aspartate substrate, it points to a biochemical traffic jam. The molecule isn't entering the cells properly, indicating a metabolic transport block. Then you have the carpenter, aspartate aminotransferase, or AST. This is the intracellular enzyme that processes the wood, and it belongs strictly inside liver and heart cells. A spike of the AST enzyme in the bloodstream means those cells have physically ruptured. It is a distress flare confirming active tissue necrosis from a heart attack or liver failure. Failing to specify which parameter the laboratory should measure leads to a diagnostic error. You risk confusing a collapsed building with a simple traffic jam. To avoid these errors, laboratories use liquid chromatography tandem mass spectrometry. This machine isolates free amino acids with absolute atomic mass precision. Despite this internal technology, the machine's accuracy is entirely at the mercy of human handling before the sample ever reaches the door. The first vulnerability is the warm tube. If a blood sample sits at room temperature, a separate molecule called asparagine undergoes a spontaneous chemical reaction. It drops an amine group and chemically shapeshifts into aspartate. By the time that warm vial is scanned, the machine registers a massive, artificial spike. A delayed walk to the refrigerator can invent a fake genetic disease on the patient's chart. The second trap occurs during the draw itself. A difficult, aggressive use of a syringe physically crushes red blood cells inside the tube, an event known as hemolysis. These crushed cells dump their dense internal reserves of AST and aspartate into the plasma, triggering a false heart attack distress flare. This is why clinical pathology enforces uncompromising protocols. Blood must be drawn into chilled tubes, placed immediately on ice, and centrifuged within exactly 30 minutes. The most advanced mass spectrometer on the planet is clinically useless if the physical chain of custody fails at the front desk. Diagnostic stewardship is vital. Physicians must reserve these complex panels for tracking severe metabolic defects and acute neurological injury, rather than screening for generalized fatigue. When an actual crisis hits, like an acute stroke, doctors utilize targeted hypothermia protocols. Actively cooling the brain lowers metabolic demand and physically slows the destructive enzymes unleashed by the excitotoxic cascade. Aspartate is a rigid structural anchor, a cellular energy smuggler, an excitatory switch, and a sensitive laboratory analyte. Clinical accuracy depends on understanding both the molecular behavior of the analyte and the strict logistical chain required to preserve it.